In this video, we will begin our discussion of the physical methods of microbial control, including heat-related methods of moist heat and dry heat, and a discussion on refrigeration and freezing, desiccation, and lyophilization. Heat is the most prominently used technique of the physical methods of microbial control. We have previously discussed the ramifications of temperature changes in Unit 2 and Unit 3. High temperatures are detrimental to the structure of proteins, nucleic acids, and lipids. In Unit 3, we learned that pathogens are mesophilic, meaning that they thrive between 20 degrees Celsius and 40 degrees Celsius. As temperatures rise above the microorganism's maximum temperature, proteins and nucleic acids begin to denature, thereby disrupting metabolism, and lipids become too fluid within the cell membrane to continue to maintain the cellular contents. Thus, high temperatures that exceed the microorganism's maximum growth temperature are microbicidal. Microorganisms vary in their susceptibility to heat, and we must take this into consideration when we elect to utilize heat as a method of microbial control. To sterilize using heat, we must consider the required temperature and length of exposure. The terms thermal death point and thermal death time reflect a combination of temperature and exposure. The thermal death point is the lowest temperature required to kill all microbes in a sample within a 10-minute period of time, whereas the thermal death time is the time required to kill all microbes in a sample at a set temperature. The thermal death time has been experimentally determined for microbial species that are common contaminants of heat-treated materials. These values allow us to compare the relative susceptibilities of microorganisms to heat and then determine the severity of treatment required to kill a specific population of microbes. We can also assess the effectiveness of heat sterilization by calculating the decimal reduction time. The decimal reduction time is the time required to destroy 90% of the microorganisms in a sample. The decimal reduction time is commonly employed within the canning industry. When considering the use of heat, we will differentiate between moist heat and dry heat. Techniques that employ moist heat make use of hot water, boiling water, or steam. Moist heat typically ranges from approximately 60 degrees Celsius to 135 degrees Celsius. Dry heat indicates the use of air with low moisture content that has been heated by a flame or an electric heating coil. Temperatures in dry heat techniques vary from approximately 160 degrees Celsius to several thousand degrees Celsius. Moist heat is more effective than dry heat. It requires lower temperatures and shorter exposure times to kill microorganisms than does dry heat. To help you wrap your mind around this concept, think about cooking spaghetti and garlic bread for dinner. To cook the spaghetti, you make use of moist heat in the form of boiling water. On the other hand, you use dry heat to prepare your garlic bread in the oven. You will willingly place your hand in the oven using a pot holder to arrange your cookware in the oven and can tolerate working in that environment for some period of time. On the other hand, you will not willingly stick your hand in the boiling water for any period of time to retrieve your spaghetti, but instead utilize cooking utensils. Moist heat destroys microbial cells by causing proteins to denature, which in turn permanently stops cellular metabolism. Moist heat also destroys the cytoplasmic membranes of cells. The moist heat techniques we will discuss include boiling, autoclaving, pasteurization, and ultra-high temperature sterilization. Boiling is a commonly used method. Boiling acts to kill vegetative bacteria, fungi, and most viruses within 10 minutes. Bacterial endospores, protozoan cysts, and some viruses can survive in boiling water from minutes to hours. Some hepatitis viruses can survive for up to 30 minutes, and some endospores survive for more than 20 hours in boiling water. Boiling is not considered a means of sterilization, but rather as a means of disinfection. It is also important to recognize that water boils at lower temperatures at higher elevations. Therefore, the boiling time is what is important. One would have to boil for a longer period of time at a higher elevation to accomplish the same antimicrobial effect accomplished in a shorter period of time at sea level. Boiling water is useful for items such as disinfecting drinking water, baby bottles, and restaurant tableware. The temperatures needed to sterilize must be higher than that of boiling water. When pressure is applied, the temperature at which water boils increases. Normal atmospheric pressure is 15 pounds per square inch. At normal atmospheric pressure, water boils at 100 degrees Celsius. An increase of pressure to 15 pounds per square inch above normal atmospheric pressure causes the boiling temperature to increase to 121 degrees Celsius. The pressure is not what kills the microorganisms, but rather the steam that is the same temperature as the boiling water. To achieve these temperatures and pressures, a device known as an autoclave is used. An autoclave has a pressure chamber, pipes that introduce and evacuate steam, and gauges that monitor the temperatures and pressures within the chamber. Sterilization is accomplished as steam condenses against the objects within the chamber and then raises the temperature of the objects, killing microorganisms on their surfaces. Thus, it is important that steam be able to contact all contaminated liquids and surfaces. The time required for sterilization is dependent on the size of the load to be sterilized. The more items within the load, the longer the time required to achieve sterilization. To ensure that sterilization is accomplished, an indicator is utilized. In some cases, a tape that is impregnated with a color indicator may be used. A piece of the tape is placed on an item to be sterilized. After autoclaving, the color indicator will appear if the appropriate pressure and temperature was achieved. In the medical setting, biological indicators are used. A biological indicator consists of a flexible plastic vial that contains a strip impregnated with Bacillus stereothermophilus and a glass ampule of nutrient media with a pH indicator. After autoclaving, the vial is squeezed to break the glass ampule containing the nutrient medium. If sterile conditions were not reached, then endospores will germinate when exposed and begin to utilize the media, resulting in a color change within the vial. If there is no color change, the endospores were destroyed by the conditions created in the autoclave. Bacillus stereothermophilus is a heat-resistant endospore-forming bacteria. If pressure and temperatures within the autoclave successfully destroy the endospores and the sterility indicator, it is accepted that sterilization was accomplished. Items sterilized using autoclaves must be able to tolerate high heat and moisture. Autoclaves are used to sterilize laboratory media and glassware, metallic instruments, and heat-resistant plastics. Items that are heat labile are destroyed in the autoclave. Therefore, if they are to be used again, autoclaving is not an option. The autoclave cannot be utilized to sterilize powders, as the steam would ruin such items, or substances that repel moisture such as oils and waxes. Pasteurization is a technique in which heat is applied to a liquid to eliminate pathogenic microbes. Pasteurization may be used for milk, fruit juices, beer, wine, yogurt, and ice cream. The goal is to reduce the microbes that would cause disease and those that would potentially cause spoilage. A number of heat-resistant or thermoduric microorganisms are capable of surviving pasteurization. Therefore, this technique is not a means of sterilization. All products have different pasteurization times and temperatures. Several methods have been employed for the pasteurization of milk, including the batch method, which heats milk to 63 degrees Celsius for 30 minutes, the flash method, which heats milk to 72 degrees Celsius for 15 seconds, and ultra-high temperature pasteurization, which heats milk to 134 degrees Celsius for 1 second. The flash method is the more commonly used method of pasteurization because it is not only effective in eliminating pathogens, but also reducing the microbial count to a level that allows the milk to be preserved during refrigeration. Be aware that these techniques do not eliminate all microbes. Thus, the products must be refrigerated to slow the growth of remaining microorganisms and the spoilage that may be caused by these microorganisms. Ultra-high temperature sterilization is accomplished by heating the liquid to 140 degrees Celsius for 1 to 3 seconds, followed by rapid cooling. This rids the liquids of all living microbes. The sterilization of the liquids allows them to be stored at room temperature for several months without microbial spoilage. This method of processing milk is popular in Europe and necessary in developing countries. It is also used for dairy creamer that is packaged as single servings for restaurants. Dry heat requires much higher temperatures and exposure times than moist heat. Dry heat destroys microorganisms by denaturing proteins and leads to the oxidation of molecules within cells. At very high temperatures, dry heat causes the oxidation of cells, literally burning them to ashes. One form of dry heat is incineration. Incineration uses a flame or electric heating coil to heat air. Bunsen burners used in laboratory settings accomplish incineration. As the inoculating loop is passed through the flame of the Bunsen burner, microbes are incinerated. Incineration may be used on a larger scale to dispose of contaminated paper cups, bags, and dressings. Ovens may also be utilized to accomplish dry heat sterilization. Circulated air within the oven transfers the heat to the materials in the oven. Temperatures range from 150 degrees Celsius to 180 degrees Celsius for 1 to 4 hours, depending on the circumstances. Dry ovens are useful for sterilizing powders, oils, or other items that may be damaged by exposure to moisture. We just completed our discussion of microbicidal techniques that employ high heat. Low temperatures, on the other hand, tend to be microbistatic. Cold temperatures only act to slow the metabolism of most microbes. Methods that utilize low temperatures are not a means of disinfection or sterilization. There are some microbes, such as the cyst of tapeworms, that can be killed by cold temperatures. However, many bacteria, fungi, and viruses are not harmed by long-term storage at cold temperatures. In fact, in Unit 3, we discussed the processes of refrigeration and deep freezing as means of preserving microorganisms. Some notable pathogens that can grow in cold temperatures include Staphylococcus aureus, Listeria, Clostridium species, and Streptococcal species. Microbial growth may be inhibited by desiccation, which may also be referred to as dehydration or drying. Microorganisms require water for metabolism. Dehydrating cells may stop the metabolism of some cells, while others become metabolically inactive but remain viable for many years. Microorganisms such as Streptococcus pneumoniae, Treponema pallidum, and Neisseria gonorrhea are all susceptible to air drying and die within a few hours. Other microorganisms, such as bacterial endospores, mycobacteria, and fungal spores, are resistant to drying. Desiccation has been used for many years to preserve foods such as beans, fruits, grains, and nuts. Lyophilization, as we saw in Unit 3, is a method utilized to preserve microorganisms for decades. This method employs rapid freezing followed by exposure to a high vacuum to dry the cells. Some cells die during the process, but enough remain viable, allowing the sample to be reconstituted many years later.